everyone. In today's session, we will talk about the next topic which comes in the molecular basis of inheritance, that is Human Genome Project, right? So, in NCERT, the next topic is 6.9. What it is? It is Human Genome Project. It is Human Genome Project. Again, a very important concept. This is again a very important concept and high-end concept also. You need to understand it in detail. So then only you will get complete clarity on this concept, Human Genome Project, right? So we tell that man is advanced. Humans are advanced. Now, when an advanced organism like human shows how many variations do you think? They show so many variations, and the scientist fraternity, they want to find out like what made the man superior. What made the man superior? They want to see the genes, their sequencing, and also then they came out with a idea like we will do the sequencing of the human genome. So first of all, if we have to start the topic Human Genome Project, we should tell that the sequence of bases in the DNA will determine the genetic information of an organism. So the biology will tell what it is. It is biology. zoology, it is zoology. In the same manner, what alphabets are there? Genetic alphabets, what are they? Biochemical alphabets. So A, T, G, C. So like that, there's a sequence. We make it as a sequence. The sequence of bases in the DNA will determine the genetic information present in an organism. That the scientists know. What they know is the sequence of bases. The sequence of bases. Where are the bases present? In DNA. What they will do? Will determine. They will determine the genetic information. They will determine the genetic information of an organism, isn't it? They'll determine the genetic information of an organism. So, the sequence of bases on the DNA will determine its genetic information. In simple, if you have to tell, the genetic makeup of an organism lies in the DNA sequence. In simple, you have to tell, we can tell that the makeup, what we are made, depends upon the DNA sequence only. So, in simple words, if you have to tell, means that is the genetic makeup. The genetic makeup of an organism. The genetic makeup of an organism depends. What does it depends? It depends on the sequence of bases. Sequence of bases where? In the DNA. The sequence of bases in the DNA. That scientists understood. Then again, RDNA technology, gene cloning, genetic engineering, biotechnology. So these areas, new areas of biotechnology. What are they? Genetic engineering. What an engineer will do? They'll give a better plan to construct roads and all, to construct bridges and all, to construct dams and all, isn't it? In the same manner, what will a genetic engineer do? They'll manipulate the genes and they'll construct the genes in a better manner so that they can express better. So with the help of genetic engineering, it is possible to isolate the DNA out, clone the DNA also, sequence the DNA also very fast. So genetic engineering techniques help the scientists to isolate the DNA, take out the DNA, clone the DNA, get hundreds and thousands of copies of your gene what you want, and to sequence the DNA also very fast. So we'll write on with the help of, what is that science called? I told genetic engineering. With the help of genetic engineering, so it is possible. What it is possible for us? With the help of this new era called genetic engineering, it is possible to isolate the DNA. We can isolate the DNA. After isolating the DNA, you can clone the DNA also, make many copies of that. We can clone the DNA also, and you can even do the sequencing. DNA sequencing also can be done. DNA sequencing can be done very fast. It can be done very fast. So the genetic engineering has opened the door to isolate the DNA, to clone the DNA, to sequence the DNA. So having knowledge about all these things, then all the scientists all throughout the world, they want to start a project, a very ambitious project. So it's a very ambitious project. Why are we telling it is a ambitious project? Okay, a big project. Why are we telling? Because if you are just thinking one person genome only, don't think that simple it is to do the sequencing of one person. It is a mega project, right? So it is a mega project. It is a mega project. Now, who took the initiative of starting it? So it is the US. So US has launched a project. US launched the project. When did the US launch the project of human genome sequencing? They launched the project in the year 1990. They launched the project in the year 1990. Understood? Why are we considering Human Genome Project as a mega project? Okay. So if I just tell the aims of the Human Genome Project, then you can understand why did we consider it as a mega project. So let us write down the aims of the project. What are the aims of the project? So the first aim of the scientists is to find out, they know that there are if you have to write out aims of the Human Genome Project, we can tell that what is the size of the human genome? We already know it. In molecular basis starting itself, we told that the length of the DNA depends upon the length of the nucleotides. The number of nucleotides in human DNA is 3.3 into 10 power 9 base pairs, haploid DNA, right? So we know that the size of human genome is how much? The size of human genome. How much we know? It is roughly 3 into 10 power 9 base pairs. We know this data, we know. And scientists, they are estimating that to do the sequencing of one base, it costs $3 it seems. So for sequencing of one base pair, for sequencing of one base pair, what is the cost they estimated? The cost is $3. It is $3. So this is an estimated cost. This is the estimated cost. Now quickly estimate for one base pair. If it costs $3, the number of base pairs are 3 into 10 power 9 bases. That means the estimated cost to complete the project for one person's DNA, it is $9 billion it seems. So then the estimated cost is, what is the estimated cost? The estimated cost is $9 billion Now will you agree? It is a mega project then? It is a mega project then. In other way also we will think. Now, whatever you have done the sequencing, whatever I am writing on the board, whatever you are understanding it, you are writing in your notes, right? In the same manner, whatever we do the sequencing, we have to store it in the books, right? Now, if you think that you are storing the information in the books, if we are storing the information, what we have sequenced, if you are storing the information in books, now, if you take a book, think that one book is having how many pages? It is having 1000 pages, right? One book having 1000 pages, and on one page, think that we will write 1000 bases. One book having 1000 pages, one page, how many bases are we writing down? 1000 bases we are writing. Then one page or 1000 bases if you are writing, you need 3300 such books. See, you need 3300 such books to store the data. We can't handle 20-30 of your books itself. You need to handle 3300 books, that too fully loaded books. That means how much data you have to store? Enormous data you have to store. That means you need to store enormous data, right? So take the screenshot if you want. Then we'll continue. What did we start in our today's class? We started with a new topic, that is Human Genome Project. So the sequence of bases which are present in us, it is our genetic information. In simple, we can tell that So the genetic makeup of an organism depends on the person's base sequence. Then with the help of a new technique or a new science called genetic engineering, very easily we can isolate the DNA, very easily we can clone the DNA, very easily we can sequence the DNA. So taking the help of genetic engineering, scientists thought that why can't we do the sequencing of human genome? So then that idea came, immediately they started, and the US announced that in 1990 it announced that we are going to do the sequencing of human genome, right? So then, and they told it's a mega project. So if you want to understand why is it a big project, mega project, so then we are looking into the aims. What are the aims of the project? The first aim we told, if you see the size of one human genome, haploid that too, 3 into 10 power 9 base pairs, and the cost for sequencing one base pair is $3. Then the cost for 3 into 10.9 base pairs will be $9 billion. That's an estimated cost. It is huge, right? The second one, if you want to store the information, we have to store the information. If we have to store the information in book, a book having 1000 pages, a page where we are writing 1000 bases, how many such books we need? 3300 such books you need if you want to store the data. That means the data is enormous. Take a screenshot, then we'll continue. Right. So for storing such enormous data, so there is a requirement of storing it in the form of databases. And we need the help of a computer, right? We need the help of a computer to store the data, to retrieve the data, right? So storing this enormous data necessitated the use of high-speed computers. So the third aim we are telling, storing this enormous data, it necessitated the use of or the development of high-speed computers. High speed, we took the help of computers also. For what purpose we took the help of computers? To store the data, to retrieve the data whenever you want. At that particular sequence we can retrieve the data, and the computer will also help to analyze the data. So we took the help of high-speed computers to store the data, to retrieve the data, and to analyze the data. Okay, high-speed computers to store the data, not only to store the data whenever we want to, retrieve the data also, to take out the data means it's called retrieve the data also, and to analyze the data also. Computer can analyze and it can tell that this much similarity is there, this much dissimilarity is there, it can help us. So we took the help of computers to store the data, to take out the data, to analyze the data. That means we are bringing up a new science where biology is merged with technology. So a new era of biology has emerged in 1990s. Okay, so then a new era of biology has emerged. You know what is that called? What is that branch of science called, which helped to do Human Genome Project? Bioinformatics. What is that called? It is called bioinformatics. Now this, what is bioinformatics? It is a new science where the computers will do programming in terms of science, in terms of biology. So this bioinformatics was developed. It was developed. So looking at all these points, can we say, or will you agree that it's a mega project? Yeah, you have to. So $9 billion not a single country can handle. And storing it in 3,300 books without any errors, that is also humanly not possible. And then they thought, like, we'll build up a computer, that area where the computer runs biological programs is called as bioinformatics. Okay, so now if you see, like 1990 US has started, right? So Human Genome Project, actually it is a 13 years project. It's a 13 years project. And when did they launch it? 1990 they launched it. In 1990 they launched it. US they launched it. So what are the major organizations they contributed? Means US Department of Energy and National Institute of Health, they coordinated first. So in 1990, two organizations, one is US Department of Energy is the first organization, and the second one is National Institute of Health. National Institute of Health. US Department of Energy and the National Institute of Health, these two coordinatedly they started the project. So what are these two have done? They have coordinated to start the project. Children, when did they start the project, Amma? They started the project in the year 1990. They started the project in the year 1990. Later, one UK organization, Wellcome Trust, came and joined them, and it has contributed a lot, and it became the major partner. So, later, we can tell Wellcome Trust. What is that trust called? Wellcome Trust. Of which country? It is UK. Wellcome Trust of UK joined these two, and it became the major partner. It has contributed a lot. So, Wellcome Trust became the major partner. Wellcome Trust also joined, and it became the major partner. So, not only US and UK, later some other countries also like Japan, France, Germany, China, and other countries also joined. So, later we can write down the countries. So, other countries, NCERT says which countries? One is Japan, they are telling. Other one is Germany, they are telling. Japan, Germany, China, right? Later other countries like Japan, Germany, China, France have also joined. So, India also joined. All these countries have also joined to do the sequencing of one person's genome. Okay, then they worked collaboratively. All these countries, they worked collaboratively for 13 years, and then they announced that they completed. They announced that they completed the human genome sequencing. All the scientist community, they announced that they completed the human genome sequencing. When in 2003 they made an announcement. In 2003 they made an announcement. On April 14 they made an announcement. 2003 April 14th they made an announcement that we have done 13 years project. 1990 they started. 2013 April 4th they told we have done the sequencing. 99.9% is completed, they told. 99.9% completed, they told. 99.999% accuracy, they told. See their confidence. 99.999% accuracy and 99.9% we did the sequencing, we completed it. That's what they announced in the year 2003. But if you see, we have 24 chromosomes. Males will have 24 chromosomes because 22 autosomes, 1 X and 1 Y. So out of the 24 chromosomes, the first chromosome is the biggest chromosome and the Y chromosome is the smallest chromosome. The first chromosome, which is the biggest chromosome, was not completely sequenced in the year 2003. They took 3 more years. Okay. The first chromosome, we will tell that the first chromosome is the last chromosome to be sequenced now. The first chromosome is the last chromosome to be sequenced. The first chromosome is the last chromosome to be sequenced. When did they sequence the when did they sequence the first chromosome? It's not in 2003. They took 3 more years. So, in the month of May, in 2006, they completed the sequencing of the first chromosome. So, this is an introduction. So, what are we telling? We told it's a mega project. So, and you cannot simply tell it's a mega project. You have to look into the aims to consider, is it a mega project? Then under that we told the first point, the size, the cost. $9 billion is the cost to sequence one person's genome, mega. Then the second point, we were talking about the maintaining the thing, 3,300 books. Again, it's not a small thing. It's again a big thing. And third thing, you need the help of computers. right? Like what are the computers, high-speed computers we need to store the data, to retrieve the data, to analyze the data. Then a new era of science has emerged. What is that called? Bioinformatics was developed. Then we were talking about, they announced it in 1990. They told we can complete it in 13 years. Then who initiated it? It's a coordinated efforts of two organizations. One is US Department of Energy. The second one is National Institute of Health. They started children. After that, who joined? Wellcome Trust of UK. These two are US departments and this is UK department joined, became the major partner. Later, other countries also contributed to their level best. What are other countries? Like Japan, France, Germany, China have also contributed to do the sequencing. Then finally, one day after 13 years, in the year 2003, they told, yes, we completed. So we completed the sequencing of the human genome. How much? 99.9% we did the sequencing with 99.999% accuracy. We completed it, right? So, but then that 0.1% which was not sequenced by the time is the first chromosome, which is the biggest chromosome. When did they complete doing the sequencing of the first chromosome? 3 more years they took, right? It's in the year 2006 May. They told, yes, we completed the biggest chromosome, the first chromosome, which is the last chromosome to be sequenced in 2006. Now, have a screenshot of this. Then we will talk about the goals of the Human Genome Project. Right? Now, when we have to talk about the goals of the Human Genome Project. So, when you are starting anything or when you joined, you have a goal, right? And you will work towards fulfilling your goal, right? You will fix your target and you will work. So, in NEET, my target is 650. So, like that, you need to fix your target 700 and you need to work out. So, like that, what are the goals of the Human Genome Project? Let's see. What are the goals of the Human Genome Project? So, we know the size of the Human Genome Project is 3 into 10 power 9 base pairs. This 3 into 10 power 9 base pairs of raw materials of the DNA bases will code for how many genes you know? Gene means a word as a gene which codes for a protein. So, they estimated that there are 20 to 25,000 genes in human genome. So, then they want to find out 20 to 25,000 genes which are present in human genome. That's their first goal. What is their first goal? To identify. It is to identify 20,000 to 25,000 genes. To identify 20,000 to 25,000 genes of human genome. This is their first goal. Right? So, if I am taking a class for 100 students, I need to identify them, their names, okay, their performance I need to identify. In the same manner, when you are planning to do the human genome sequencing, you need to identify there are 20 to 25,000 genes, you need to identify them. That's their first goal. Then their next goal is to determine the sequence of all this 3 into 10 power 9 base pairs, right? So, what is the sequence? Biology, if it is a DNA, the sequence is BIOLOGY. In the same manner, they want to determine the sequence of all the 3 billion chemical base pairs of the human genome. They want to determine the base sequence. We need to know the base sequence of 3 into 10 power 9 base pairs. If you simplify, means it will come 3 billion chemical base pairs. If you simplify 3 into 10 power 9 means you will get 3 billion chemical base pairs, right? So, they want to do the sequencing of 3 billion chemical base pairs of human genome. Correct? Now, if I have to correct your biology paper, first I should know the concept correct, then only I can make out that you are making mistakes. In the same manner, if you want to study the genomics, if you want to study human genetics, first of all, you should know what is the order. Then only you can identify that there is a mistake or mutation happened here. So, that's why the second goal is to determine the sequence of all the 3 billion chemical bases of the human genome. And the third one is, so we take NCERT textbook as a reference and we study it. So, in the same manner, you need to store the information somewhere, right? Where? Computer, in the databases. Whatever the scientists are doing, they have to store it in the form of databases. That's the third goal, right? So, they need to store the data. They need to store the data. Where? In databases. They need to store the data in the databases. So later on, when any scientist want, so it's immaterial, like this country has done it, this country has done it, anyone can retrieve the data. So that is their third goal. Now fourth one is they want improved tools for data analysis. So they want improved tools for data analysis. What do you mean by improved tools for data analysis? Means, for example, I am interested to do research on insulin gene. I should know that the insulin gene is on the 11th chromosome on the longer arm, on the P arm it is there. I should know that, right? So then I need not start from chromosome 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. I should not reach the 11th one. So if I can retrieve the data, if I can analyze the data, straight ahead I can go to the 11th chromosome. Straight ahead I can pick the sequencing, I can pick the gene which codes for insulin. That's what is called improved tools of data analysis. The next one is to transfer the related techniques to the other sectors also, is to transfer the related techniques. Transfer the related techniques to other sectors also, like industries. Actually, these research topics are done by institutes. This research is done by institutes, but if you see, hardly how many institutes will be there, and hardly how many are potent enough to do that. So if you transfer it to the industries, then they have funding and they can finish the data more faster in a better manner. So transfer the related technologies to the other sectors like industry is their next aim. And then the last aim, the last goal will be to address the ethical, legal and social issues. So in short form we will write ELSI, ethical, legal, social issues. Now we tell that man is superior, man is superior, man is on the top. Now and then we tell that like it happened like this, man is so much evolved. Now when you want to decode the complexity of the man, you know, so people will come and they raise objections. Ethically they'll object, legally they'll object, socially they'll object. Then you should be in a position to address all the issues that arise from the project. So address the ethical, legal and social issues that arise from the project. So children, these are their goals. How many? 6 goals they have kept for the project. What are the 6 goals? Come on, tell me. The first goal is to identify the 20,000 to 25,000 genes which are present in the human genome. Like 25,000 students are there in the class, means I need to identify. So that is my first goal. Now the second goal is to determine the base sequence of all the bases which are present. Like 3 billion base pairs are there. So these many students are there. I want to know their names individually, like that. To determine the base sequence, biology is a gene. The sequence of biology is BIOLOGY. Like that, this is a human gene. What's the first base? What's the second base? What's the 10th base? What's the 100th base? They want to do the sequencing also. That is their second goal. Third goal is to store the data. Whatever they have done, it's not their work. It's a collaborative work. They need to store it. That is their third goal. And the fourth goal, they want to develop improved tools for data analysis. They want somewhere in between. So then the computer has to analyze and to give that much data only. They should not search for days together to reach that particular point. So improved tools for data analysis is the fourth goal. Now next one. So not only the institutes, they want to transfer the technology to the industries also, which can help them to do the sequencing fast. That is their fifth goal. And the last goal is to address. So when you are doing anything like ethical issues, they will rise, legal issues, they will rise, social issues, they will rise. So you should be in a position to address them, to address the ethical, legal and social issues. So these are the goals. So far what we have seen why it's a mega project. Then we have seen who contributed it. Then we have seen what are the aims. Then we have seen what are the goals. Now, by having a knowledge about these DNA variations, So, having knowledge about DNA variations. If you have the knowledge about DNA variations, then it will lead to a revolutionary new way to treat a disease, to diagnose a disease, and it helps in prevention also. When you know the DNA sequence, for example, if I know the biology spelling is BIOLOGY and mutation happened and it became something like CIOLOGY, then you understood, you can easily diagnose it and you can easily treat it also. Gene therapy will replace C with B again, will bring the B back again. So it helps in diagnosis, it helps in treatment also, it helps in prevention also. So knowledge about this DNA sequencing, what it will do? It will revolutionize. It will revolutionize the new era of gene diagnosis. You can diagnose it, you can treat it, and you can even prevent it, isn't it? We took an example, simple example too. If you know the sequence, you can make out here is a difference, this gene is a defect, we can treat it and we can prevent it also. Understood? Now, beside providing clues of human biology by doing the sequence, we can tell that besides providing the clues of human biology. So, till the Human Genome Project was done, we used to think that human is the ultimate power and like this it happened. But now we came to know, okay, this is a gene, it codes for this, there's a defect here, now it runs in the families, this is dominant, this is recessive, everything we understood. Every clue we got about human genome now after doing Human Genome Project. So, beside providing clues of human biology, now these scientists, not only they did the sequencing of human biology, they have also done the sequencing of non-human models also. So, sequencing of non-human models. Now, what are the non-human model sequencing they have done? Now, if you want to get into medical, straight ahead you cannot go take your bag, sit in a medical college. You need to finish your pre-primary, then primary, then your school education, then your 12th education, then you need to then score a rank in NEET, then you can get into medical college. Like that, there's a way. In the same manner here also, you cannot directly do the sequencing of a high level man, right? So first we should start from a bacteria which is a prokaryote. From a unicellular bacteria, we will go to unicellular eukaryote like yeast. Then we will go with a nematode. Then we will go with a plant. Then they have come to animal. So, before doing the sequencing of humans, they have worked out the sequencing of non-human models like, a clever person will always do with a unicellular prokaryote, that is bacteria. They have done it. They succeeded. Then, okay, let's go on to the unicellular eukaryote. They have done with the yeast. They succeeded. Then, the third time they have done the sequencing of a nematode, free-living non-pathogenic nematode. It is Sinorhabditans elegans. Sinorhabditis, ELEGANS. This came in the NEET previous exam. They asked, what is the Sinorhabditis elegans? It is free-living, non-pathogenic nematode is the answer. It is free-living. It is not disease-causing, so it's called non-pathogenic. And what it is a nematoda. It's a nematode. They asked, what is Sinorhabditis elegans in 1 year of the NEET paper? It is free living, it's not parasitic, and it is non-pathogenic also. And what it is nematode. First they have done with unicellular bacteria, it worked out. They have done the sequencing of yeast, it also worked out. Then they came and did the sequencing of Sinorhabditis elegans. After doing the Sinorhabditis elegans, they came to an insect, that is Drosophila. Fourth one, they have done the sequencing of Drosophila. Then they came to the plant. In plant, first they have done the sequencing of rice. After rice, they went and did the sequencing of a weed called Arabidopsis thaliana. Arabidopsis, Arabidopsis thaliana. So, this is a weed, monocot weed. They have done the sequencing of Arabidopsis thaliana. They have done the sequencing of rice, 1, 2, 3, 4, 5, 6. All these 6 non-human genome models they have sequenced. Before doing the sequencing of the human, they understood the complexity, they understood the makeup which helped them to do the sequencing. So by doing the sequencing of all these things, it helped them to solve some areas in health care. It helped them, doing the sequencing of these things, it helped them to work out in healthcare, work out in agriculture. It helped them to work out in new techniques in agriculture. It helped them to work out in bioremediation. You know what is meant by bioremediation? Clearing the nature by using microorganisms is called bioremediation, environmental bioremediation, and energy production also. So by doing the sequencing of all these things, they worked out this sequencing. They cannot use it for Human Genome Project. So they worked out this sequencing in healthcare, in agriculture, in environmental bioremediation, and in energy production. So in this board, we talked about the goals of the Human Genome Project, and then with this knowledge, what we can do, and then what are the non-human genome models they have done the sequencing. 1, 2, 3, 4, 5 and 6. Where did it help? In 4 areas. 1, 2, 3 and 4. So take a screenshot. In the next lecture, we will continue the remaining part of this topic, where we will talk about the methodologies of Human Genome Project, salient features of the Human Genome Project, then the applications of the Human Genome Project. Half the way we completed. In the next lecture, we will talk the remaining part. Hope you like the lecture. So if you like the content, like, share and subscribe to my channel. Thank you.